This is Mrs. Palmer-Quay with the first video for Unit 7. This video covers all of the learning targets in Section 1 and gives you an overview of the nervous system, including its general structure and function. We can start off by dividing the nervous system into two divisions. There is the central nervous system, which is composed of the brain and the spinal cord, and that's more centrally located on your body. This is abbreviated often just CNS to avoid having to write out central nervous system so many times. And then there is the peripheral nervous system, which involve the nerves, the cranial and spinal nerves that go to the rest of the body. And it also has the abbreviation PNS. You can further divide the PNS, the peripheral nervous system, into two sections. There is the sensory division, the part of the nerves that are providing us information, sensing what's going on in the environment, externally or internally in our own environment, and the motor division, the nerves that provide some type of motor reaction. The motor division can be further subdivided into the somatic nervous system, and in the somatic nervous system you have the motor neurons that go to skeletal muscle, like we were studying in the last unit. And then you have the autonomic nervous system. Autonomic is an awful lot like automatic, and so hopefully you can remember that these are the nerves that go to smooth muscle, cardiac muscle, and glands. They are reacting in response to nervous stimuli. They react automatically. We don't have to think about keeping our heart beating or excreting a particular hormone. Our autonomic nervous system takes care of that automatically. Furthermore, you can subdivide the autonomic nervous system into the sympathetic and the parasympathetic divisions. And we'll talk about these a little bit more when we get to the peripheral system in several videos from now. But in general, the sympathetic division is involved in reactions that are used when you're in a fight or flight situation. This sympathetic division gears up when you are stressed and need to make a decision and need to make a movement. And so it then is in contrast to the parasympathetic division, which gives you times for rest and relaxation. So the sympathetic tends to gear up your body and the parasympathetic tones it back down again. There are three major functions of the nervous system. It is involved in the sensory function or receiving input. Sensory receptors are in your peripheral nervous system. They are at the ends of your peripheral nerves, and they carry information to the central nervous system. They are also known as the afferent neurons. Then you have the integrative or the deciding part of your nervous system. And in the integrative part of your nervous system, the sensory information is taken in and evaluated. And this happens, of course, happens in the central nervous system. It's taking in sensory information coming from the peripheral nervous system. So in the integrative function, your central nervous system, and primarily your brain, is processing, thinking about, analyzing, determining what you are actually sensing by all of this stimuli. The neurons that do the integrative function are known as interneurons or association neurons. And I see I missed an I in here. Sorry about the misspelling. Association neurons. And then third, there is the motor function of the nervous system, and this is where you are reacting to the information that your integrative system has, or integrative function has dealt with. And in the motor section, nerve impulses again travel in the peripheral nervous system. They are traveling from the central nervous system out to muscles and glands. These are known as efferent or neurons because they make different effects happen. So that you receive information, you make a decision about this information, and then you react to this information. These are the three functions of your nervous system. The neuron, as I hope you remember from the unit on tissues back in the beginning of the year, the neuron is the basic functional unit of the nervous system, and it has three basic parts. There are some different shapes to neurons, and we'll get to them in a minute. But basically, all neurons have a cell body, and that is the cell part of the neuron with all of those cellular functions that you are very familiar with. So there's mitochondria in there, there's cytoplasm, there's lysosomes, there's endoplasmic reticulum, ribosomes. All of those organelles are found in the cell body. Coming out of the cell body are some to many branching little extrusions of the cytoplasm, and those are known as dendrites. And one of those dendrites is extra long, and it has a special name. It's an axon, and it has usually some branching at the opposite end, at the terminal end. The transmission of nervous impulse always goes from the dendrite end down the axon, so the nerve impulse always travels in this direction. It doesn't ever flow backwards. And to help it flow quickly, many of your neurons are covered in a substance called myelin, and we'll get to how that happens in a minute. But the myelin sheath coating of your axons actually means that the impulse can jump quickly from in between one section to another, and things can be transmitted much faster down the neuron. So here are some of the shapes that you can see with neurons. The multipolar neuron, which you saw on the previous page, is the most common version, primarily because it is found in the brain, and we've got so many neurons in our brain, it overwhelms the numbers in any category. They can be heavily branched, much more branched than it's even shown on this simple diagram, as neurons connect to other neurons in the brain to allow us to make memories and to think about things. The whole idea of consciousness is still barely understood. We really don't understand how our brain works. And then there are the bipolar neurons, and these have a cell body in the middle with sort of a long dendrite off one side and a long axon off the other. These are found in some specific sensory organs. They are found in our retina, helping us process the information about how light comes in the inner ear, and in the far regions of the nasal cavity. So these are involved in some of the sensory functions, these bipolar neurons. And then the unipolar neurons, which have the cell body in the middle, and then from the cell body there only is one process coming out, which then divides in two to form the dendrite and the axon. Instead of the unipolar or the bipolar, you've got a process coming out on both sides of the cell body. The unipolar, you just have one processor coming out and then it divides. And many of our sensory neurons are unipolar. Sensory receptors, we talked a little bit about specific receptors in the integumentary system, but there's another way of dividing them into the sensory receptors based on the nature of the stimulus that gets them excited. Because here's where we're talking about things that are exciting our nerve cells so that a nerve impulse will be formed and travel down the nerves. So what types of things excite nerve cells? We have receptors that respond to changes in temperature, thermoreceptors, and these are receptors that change in the general room temperature sort of range, not extremely hot or extremely cold, but things that become warmer or colder in sort of what's known as the innocuous range. They're not going to cause damage. Innocuous means harmless. So we have both warm or warming receptors and cooling receptors. One receptor doesn't handle both temperature changes, but you've got it for both ways. Things are getting warmer or things are getting cooler. Two different receptors there. You have mechanoreceptors. These respond to changes in mechanical pressure as opposed to atmospheric pressure, or to physical distortion of body parts. So stretch receptors that respond as muscle and body parts are stretched as you moved are a mechanoreceptor. Chemoreceptors are sampling the chemical concentration either of things in the gaseous state, like your sense of smell is picking up molecules that are traveling in gaseous state, or in aqueous solution, and they may be involved in not only picking up sensory information from the outside environment, but also very much internally when it comes to all of the homeostatic processes that are going on in your body, maintaining your homeostasis by monitoring your blood carbon dioxide level or the level of a particular ion. Those are all handled by chemoreceptors. And nociceptors are a special type of receptor that's activated by tissue damage, and they generally then is perceived by our brain as something painful, but the nociceptor may be responding to an extreme change of temperature, and so it comes out in pain, but it's actually a thermoreceptor nociceptor, so it has sort of subdivisions that fall in each of these categories, or an extreme amount of pressure, a very strong poke, or a toxic amount of a chemical may actually cause pain. So the nociceptors actually are subsets of these various other categories because of their ability to be activated by extremes in each of those other categories. Then along with your neurons, the neurons that provide the nerve impulse and the electrochemical signaling, you also have neuroglia cells. And neuroglia cells are actually much more present than neurons. There are many, many more neuroglia cells than neurons in your nervous system. But they are not involved in transmitting your nervous impulse. They are involved in supporting and protecting and taking care of your neurons. There are 4 types of neuroglia cells that are found in your central nervous system and 2 types found in the peripheral nervous system, and we want to look at those specifically next. In your central nervous system you have, and this of course is inside the brain or the spinal cord, these are the 4 types of neuroglia cells. You have the microglia or microglial cell, and it is a cleaning up cell. It will be involved in traveling, and by phagocytosis will clean up any bacteria that may be in there, any dead, damaged tissues, keeps things tidy if there's an injury, trauma to the central nervous system. The microglial cells are in there to clean things up and then also to form some of the scar tissue that may happen because of repair. You have the astrocyte, which is star-shaped, which is why it has this particular name. And you see it here wrapping around a capillary. One of the very important things that astrocytes do is provide the blood-brain barrier. They are keeping substances from traveling out of blood vessels and into the brain. The brain is much more sensitive to chemicals and toxins than the rest of the body, and so the astrocytes keep things from moving easily out of the blood vessels into the brain. This can be a challenge in getting drugs to the brain if there should happen to be something like an infection in your brain. Doctors have to use other methods. They can't just give you an injection and expect your blood to carry it around. But that's because the astrocytes are providing this blood-brain barrier. They're also very much involved in embryonic development, helping the brain cells, get to the right place. Then inside your brain there is some space. They're known as ventricles. Down the middle of your spinal cord there is a space called the central canal, and traveling all throughout this space is your cerebral spinal fluid. And the ventricles and the central canal are lined by this epidermal cells, and they have cilia. You can see the little cilia here on the end. They look an awful lot like cuboidal epithelial cells, but they are not. So the epidermal cells are having cilia that beat and circulate the cerebral spinal fluid. They make sure then that nutrients get moved around to the different parts of your central nervous system, and they provide a lining to those 2 open cavities. And then you have the oligodendrocytes. The oligodendrocytes are involved in providing a coating on the axon, the myelin sheath, by wrapping little sections of the axon with a extrusion of their cell membrane. So basically it's like a finger comes out, it sends out a little bit and it wraps around the cell membrane, around the axon, and slides under itself and spirals in until you have this tight coating of really cell membrane tissue around and around and around the axon in a particular place. Any oligodendrocyte maybe have 15 different of these little protrusions out connected to several axons, and so they are, as we will see in a minute, are involved in increasing the speed of the electrical impulse down the nerve cell. In the peripheral nervous system, you only have two types of neuroglial cells. The schwann cell has the same role as the oligodendrocyte in that it provides the myelin sheath. Instead of having several little fingers of cytoplasm come out from one cell, the schwann cell wraps its whole self around the axon. And so this would be one schwann cell, and this would be another schwann cell that's been partially dissected here, and here's another schwann cell. And so they, again, will slide their cytoplasm, and because they are an individual cell, they actually don't wrap in reverse like this, but they would start wrapping around the axon until we got to the outside, and then at the outermost portion you have the rest of the cell body. So there's the nucleus and all of the other functional parts of the cell kind of lumped up on the outside. It's not really drawn well in this picture, but next to the axon's running down the middle of it here. Next to the axon you have layers and layers and layers of cell membrane. Then this, if you remember back to the pictures of the different types of neurons, this is a unipolar neuron. Here's one process coming out that then splits into two directions. And so here is the cell body, and it is wrapped by these satellite cells which provide electrical insulation and protection around the cell body of the unipolar neurons in the peripheral nervous system. In the peripheral nervous system, we can have regeneration of neurons, and it is due to the schwann cells. So here is an axon, and you can see we've got all these individual schwann cells wrapped around it. There's been an injury here, and of course this part of the axon then has lost contact with the rest of the cell. It no longer has the access to all of the cellular processes that are going on in the nucleus, and so that tissue is dead. It has no way to survive. It will slowly decay and disappear. But the rest of the neuron is still alive because the cell body has not been injured. If all you have injured is the axon, then regeneration is very possible. The schwann cells are able to exert a growth factor that helps the axon regrow, and if all goes well, you will then have a reattachment of the nerve cell to the muscle fiber or wherever it was originally attached. It may take as much as 2 years for this regeneration to happen. And unfortunately, because it's sort of growing blindly here through your body, it may not attach to the muscle fiber that it originally attached to. It may not attach to a muscle fiber at all. It may never find an attachment site. And so you don't get 100% return if you're having to have a regeneration of a neuron because of tissue damage. But oftentimes you are able to get a lot of the function back. This is in contrast to the central nervous system, which does not have schwann cells, and the oligodendrocytes don't seem to be able to produce the growth factor the same way as the schwann cells do. So you don't get this type of regeneration of the axon of the neurons. Once the central nervous system neurons have been damaged, they're pretty much not able to be repaired, and you just have scar tissue develop. I experienced this personally, not from a motor neuron, but I had managed to kill off some connections to sensory neurons this spring and had sections of the skin of my thumb totally numb, but it has managed then in several months, not several years, to regain the sensation as these axons repaired themselves and developed those sensory endings again. Neurons themselves are said to have two distinct functions. They have irritability and conductivity, and that's what makes them do what they do. Irritability, if you remember back from way back in biology, talking about characteristics of life, a cell that is irritable has the ability to respond to stimuli. And nerve cells take that stimuli and convert it to an impulse, to an electrical impulse. And then they pass that electrical impulse on to other neurons or directly onto muscles or glands if they happen to be a motor neuron. So without these two functions, our nervous system would not work. But because our neurons are responsive and they are irritable and they are able to conduct these impulses onto other cells, we are able to communicate from one end of our body to the other and enjoy all of the thinking and processing ability by our brain. So let's talk a little bit about these nerve impulses. Very similar to some of the stuff we already talked about with muscles, so hopefully this will not be an overwhelming subject as we start to work through some of these specifics. Now, if you picture a cell membrane under normal conditions, so we'll just kind of put a cell membrane here, there are sodium ions in the extracellular environment, and there are sodium ions inside the cell. But there's many, many more sodium ions outside because there is a little pump in the cell membrane that constantly is moving them out. This sets up a resting potential. There's a difference in ions, much more positive out here than it is inside. So we have a positive and negative difference, like a concentration difference, but it's a difference in charges. So it becomes an electrical difference. It becomes an electrical difference of about a negative 70 millivolts, and you don't need to necessarily memorize that number. Then if something happens that allows some of these sodium ions to start to trickle back in, if a channel opens up, an ion channel opens up that has been closed, and that's of course another integral protein in the cell membrane, so if that allows your sodium ions to travel back in, you're going to decrease the potential difference. And so if you are able to get it down to a negative 55 millivolts, then you will trigger an action potential, and there'll be a falling dominoes level of activity that sends this nerve impulse down the axon. And it's all concerning whether you have sodium inside or outside the cell, what is the positive versus negative difference across the cell membrane. So here we have a diagram of a cell body and a little portion of the axon, and you can see the ends of the axons from some other neurons that have come in and connected with this particular nerve cell. And we're going to get to the fact that what is given off at the end of one axon is a neurotransmitter, is a chemical, like you saw with the muscle cells. So these little dots are showing the neurotransmitter. And what this neurotransmitter is doing to this particular neuron is that it is opening up the sodium ion channel. You see it says there's a chemically gated ion channel here, that as the chemical is picked up by receptors on the cell membrane, it opens up an ion channel and lets some sodium back into the cell. So this is going to change from the negative 70 volts, it's moving it down to a negative 62. It's adding a little bit of potential changes locally. At this point, it's not enough to trigger a nervous impulse, and this sort of thing goes on all the time, that it allows your body to sort of filter out the unimportant stimuli because it's not enough to trigger a full action potential. A nerve just sort of ignores those little impulses that come through as long as we don't reach the threshold stimulus. So in this lower diagram down here, of course, we've got several more of these sodium ion channels opening up because of the chemical stimulation from the other neurons, and it gets to the point where it does get down to that threshold stimulus of negative 55 millivolts. And so inside the very, very beginning section of the axon, there's sort of a bulge section here called the axon hillock. That's represented by this bigger section. And the sodium channels there are not chemically controlled, but they are voltage controlled. So as soon as this voltage level is reached, they open, and they open in a much larger number and allow the sodium ions to just really flood the inside of the cell, so they all rush in. So this is a slow process in the beginning as the stimulus is building, as the chemicals that are, the neurotransmitter chemicals are slowly allowing sodium in the cell, but once you hit the threshold stimulus, then you have a voltage-gated channel open up and everything suddenly happens. Here's another illustration of the same principle. So in the resting situation, the resting potential, the sodium channels are closed, and this is just a little picture of the sodium protein. And then if we just sort of jump down to right here, this is a region undergoing depolarization. The action potential is moving through the area. The sodium channels are open. Sodium ions are flooding into the cell, and it works one region at a time. So the entire axon does not get depolarized at the same time, but just a little section does, and then it passes on and triggers the next section, which then passes on and triggers the next section until it has moved all the way down the axon. After a section has been triggered, there is a brief time period where it's known as refractory. And in these cases, the sodium ion channels, you can see they still look like they're open, but they have some sort of lid on. They've been rendered inactive. And so after the action potential has passed through, that part of the membrane for a brief millisecond cannot be repolarized. It cannot move another impulse down. It is a time period where it's resting, and that in part is the reason why action potentials only move in one direction. They can't move backwards because the part of the membrane it just has passed through is not able to accept another impulse. And so this refractory period where it is not able to bring in sodium ions, it is not able to respond with a nervous impulse, is a protective mechanism that makes sure that nerves only travel one way. Then at the very end of the axon, we have the synapse. That's the space here between the axon of one neuron and the dendrite of the next. And we call this particular nerve the presynaptic neuron because it is before the synapse. And of course, then this one would be the postsynaptic neuron. But in that space, the signal changes. It is electrical coming down the nerve cell, but it is chemical going across the synapse. And you've already seen this with muscle cells, so this should be very familiar to you, that you have various vesicles that are containing a particular chemical that we call a neurotransmitter. Acetylcholine is the one that we used with muscle cells. And these vesicles then fuse with the cell membrane and release the neurotransmitter into the synapse when that nervous impulse reaches the end of the axon. The neurotransmitter then is picked up by receptors on the dendrite of the next nerve cell, and then these are the receptors that trigger the chemical gates, the chemically triggered gates that allow sodium to enter in that start to stimulate this nerve cell. In order to stop nerve cells from being continually stimulated, of course, there are enzymes that will either destroy the neurotransmitter completely, or some of them actually recycle them and bring them back into that presynaptic neuron. So the process of your nervous impulse is electrical coming down the axon, chemical across the gap, across the synapse, and then electrical again through the next neuron. Neurotransmitters are divided into two categories. They can cause an excitatory response or an inhibitory response, and the name tells it all. The excitatory response leads to depolarization. Remember, that's what we call when the membrane is ready to send down an action potential. So excitatory neurons increase the depolarization of the postsynaptic neuron, and so the action potential becomes more likely. A very common example of an excitatory neuron or excitatory neurotransmitter, because other substances besides what our bodies other substances besides what our bodies create can act as neurotransmitters. And caffeine is an excitatory chemical. It makes our neurons more likely to respond, which we interpret as feeling more awake. We respond more quickly to the stimuli in the environment. An inhibitory neurotransmitter, or another inhibitory substance that serves the purpose of a neurotransmitter, does the opposite. It hyperpolarizes. It makes the membranes appear more refractory, and so it makes the chances of an action potential being generated much less likely. The Botox treatments that people get because they don't like the wrinkles in their forehead are an example of an inhibitory neurotransmitter. And this is just the botulism toxin being given to you in very, very small amounts. It prevents the muscle cells from reacting to any kind of nervous impulse, and that leads to a lack of expression, a lack of wrinkles and contractions under the skin, which people who are concerned about their appearance find more pleasing. We mentioned already that myelinated axons allow the nervous impulse to jump from one space to the other. The space in between the myelin sheaths, and this is an example of a schwann cell, they're giving you the peripheral nervous system again, and of course this actually would have sort of a bulgy section that would have a nucleus and a cell body. But this stylized diagram is showing you a number of schwann cells, one after the other along the axon, and it's showing how the nervous impulse is going to be able to jump from the spaces in between. So the change in electrical potential only happens on these little tiny spaces in between the schwann cells. And these spaces are called the nodes of Ranvier. And because only a small part of the total membrane of the axon needs to be polarized, the impulse can be transmitted much, much faster, in fact up to 50 times faster down a myelinated axon than an unmyelinated one. So many of your nerves have this myelination around it, which allows you to transmit impulses faster, allows you to think faster. It keeps the directions that your brain is sending to your motor neurons to quick response time. The disease multiple sclerosis is an autoimmune disease that causes your body to attack the myelin sheaths on the nerve cells, starting with those in the brain. And so one of the effects, one of the immediate effects that's usually seen from multiple sclerosis is just a general slowing down of nervous impulses. People are not able to move their muscles as fast because their myelin has been lost around the muscle, around the axons. And then, of course, with multiple sclerosis, condition can get much worse beyond that. But it is a condition that affects the myelin sheaths, and then because of that, the speed at which nervous impulses are transmitted. As you saw with muscle cells, nervous impulses are all or nothing. If a neuron is going to respond at all, it's going to respond completely. And as soon as that stimulus of the threshold intensity or threshold potential is reached, then you will have an action potential. The axon moves, or the impulse carried down the axon always have the same strength. The degree of response that you get from any particular stimuli doesn't depend on a particular neuron, but it depends on how many neurons are involved in responding. So a more intense response will be because you've got more neurons sending you information. Like the muscle cells, a greater contraction is because more muscle fibers are activated. A greater nervous response is because more neurons are activated, because each response is all or nothing. It only can be at one strength. Now I want to talk just briefly about reflex arcs. These are an automatic response to stimuli that can occur either externally or internally. So if you have sneezed or coughed in response to something in the air, or once your mouth liquids get to the back of your mouth, you swallow whether you want to or not. Things that are involved in homeostasis, your heart rate or your blood pressure, these are all maintained automatically by reflex arcs. And reflex arcs may not involve these interneurons. It may not involve your brain, because it may be just a connection between a sensor neuron and a motor neuron. This is especially true of the stretch reflex arc. You generally have a little bit of connection with your brain with a withdrawal reflex, and I want to talk you through these two reflex arcs next. The stretch reflex arc is when muscle stretch, which is sensed by one of those mechanoreceptors deep in your muscle cells, the stretching of the muscle fiber causes the muscle to contract. It's very important for maintaining a balanced posture, because you don't have to think about if you tilt too far in one direction, say you're leaning to the right and the left side of your body is getting stretched. Well, the left side of your body then reacts by contracting, and it pulls you back upright. This response is what is tested by the little patellar tendon test that you may have happen at a doctor's office by pushing on the patellar tendon, and we talked about how your quadricep muscle all comes down and join together on the patellar tendon. And so it will stretch your quadricep muscle up here, and in response, your quadricep muscle will contract and your foot will kick out. This is only a reflex arc that only involves two neurons. You have your sensory-receptor neuron coming into the spinal cord, connecting to a motor neuron going back out to the muscle. There is no connection to the brain, and if you think about it, if you've experienced this particular reflex, you really don't have information going to your brain about it. You see it, and then you can think about it, but there's not sort of an internal connection saying something is happening in your quadriceps. And so the stretch reflex is something that just happens from a sensory neuron connecting directly to a motor neuron, and there's no sort of processing that happens at the same time. In contrast is the withdrawal reflex. It again has a sensory and a motor neuron connected, and it allows a quick response by your muscles to some potentially damaging situation. So this is when you feel heat, as this little diagram shows, or you prick your finger or step on a tack or something. You're withdrawing from that painful stimuli, and you do it without having to think about it, but usually because you're feeling pain, your brain eventually does get involved. And so along with your sensory neuron coming into the spinal cord and your motor neuron coming out, you usually have an interneuron in between, and it passes the information on from sensory to motor, but it also then passes it on to go up to the brain, so that the neurons in your brain can process this information and learn from the experience not to touch the hot stove or to be careful of sharp points on needles or something along this line. So that this particular reflex arc does have a thinking component because it does involve an interneuron, whereas the stretch reflex is a straight sensor to motor neuron connection. So that covers your overview of the structure and function of the nervous system. There'll be 3 more videos for this unit, each one covering one section in Unit 7.